Welcome dear students, welcome to your channel The Anatomy Canvas. In today's class we are going to discuss the cartilage. So the learning objective for the today's class is to discuss general feature of the cartilage, to discuss its component and to describe the different types of the cartilages histologically and also their location in the body. So first, by definition, cartilage is a form of the connective tissue composed of cells called chondrocytes and highly specialized extracellular matrix. So this is a specialized type of the connective tissue. It is an avascular tissue, very important point. Cartilage do not have direct blood supply. It consists of the chondrocytes and it has an extensive extracellular matrix. More than 95% of the cartilage volume consists of extracellular matrix, which is the functional element of this tissue. It supports the regions of the body that require flexibility. It is a non-nervous structure, again very important MCQ. It has a poor regeneration capacity, again very important MCQ. Usually it is surrounded by pericardium except the articular cartilages and the fibrocartilages. So these are the general features of the cartilages. As we know, these are the specialized type of the connective tissue. It is avascular, it is non-nervous, poor regeneration, surrounded by pericardium except in certain location, that is articular cartilage or the fibrocartilages, and also it consists of the chondrocyte and the extracellular matrix. So what is pericardium? First of all, the pericardium is the dense connective tissue that is composed of the cells that are indistinguishable from the fibroblast. So you can see in this slide, from this area to this area, you can see this is the pericardium. This is dense irregular connective tissue, and again this pericardium from here to here. This pericardium is divided into two layers. The outer layer is the fibrous connective tissue and inner cellular layer, which give rise to the new cartilage cells. So in this area you can see smaller cells which resembles the fibroblast. These are present over here in this inner cellular layer, and these cells are actually the chondrocytes which are present over here, and they have the capability to regrow. So this is the next area, which is the germinal or growing zone. From cells from this area divide and they form the chondrocytes and they add to the new cartilage. So pericardium is the outer covering of the cartilages except in two locations, and it consists of the two layers. Outer is dense irregular connective tissue and inner is more cellular layer. Next, what are the different components of the cartilages? So it consists of the cells. These are the chondroblast and the chondrocytes. The connective tissue fibers and the extracellular matrix are the ground substance. So we all know whenever we describe any type of the connective tissue, we have to describe it under the three headings: the cells, the fibers and the ground substance. So in case of the cartilages, the cells are chondroblast and chondrocytes, and the different types of fibers and ground substance. So we discuss all of these one by one. So first of all, the cells. These cells are differentiated from the derived from undifferentiated mesenchymal cells. They are the specialized cells produce and maintain the extracellular matrix. Here you can see these are all are the chondrocytes. When these are active, they are called chondroblasts. When they are in quiescent state, they are called the chondrocytes. So they are distributed either singly or they may be present in the form of the clusters. These clusters are called isogenous groups. So they may be present either single or maybe in the form of the clusters. So these are the chondrocytes. These chondrocytes are present in a space which is called the lacunae. So in this condition you can see these are the spaces of the chondrocytes, and these chondrocytes are present in these spaces. So the chondrocytes, they are present either singly or in the lacunae. Here you can see these are the lacunae. These are the isogenous groups in which chondrocytes are present. 2 to 4 chondrocytes are present in a group, or they may be present in the in a single form. So in both of these locations you can see these chondrocytes are present in a space which is called the lacunae, and in the center you can appreciate very well the nuclei body of the chondrocyte is also present. So these chondrocytes secrete the territorial matrix. This territorial matrix is highly concentrated in nearest to the chondrocytes, where it is also called the capsule, and the next zone to this capsule is called the territorial matrix. And the territorial matrix, the ground substance which is present in between different isogenous groups, is called the interterritorial matrix. So you can appreciate in this diagram, this is the interterritorial matrix, this is the territorial matrix, and the territorial matrix which is just close to the chondrocyte is called the capsule. So this is because of the differences which it secretes, that in a close vicinity to the chondrocyte it is highly sulfated, that is why it gives color to it, and in the areas in the interterritorial matrix it also contains the fibers of the chondrocytes, that's why it is little bit lighter in color. So dear students, this is the electron microscopic picture of the chondrocytes. Here you can see the nucleus which is present in eccentric position, the abundant dilated rough endoplasmic reticulum which is present, the Golgi apparatus present over here as well, large number of the mitochondria present in this location as well. So large amount of the dilated RER and the Golgi apparatus indicate that this cell is actively engaged in production of the cartilage matrix. So in addition to RER, Golgi apparatus, mitochondria, it also contains the secretory granules, the vesicles, the intermediate filaments, microtubules, and actin microfilaments. The numerous dark particles which are present in the matrix, it contains the proteoglycan. Particularly larger particles adjacent to the cells are located in the region of the matrix and identified as the capsule or the territorial matrix. So what are the functions of the chondrocytes? The chondrocytes secrete and maintain the extracellular matrix. They secrete collagen component. They secrete non-collagenous proteins, that is proteoglycan and glycosaminoglycans, and they secrete the metalloproteinases. So next is the extracellular matrix of the cartilage. Extracellular matrix is firm, but it also very pliable. It consists of three main constituents: the collagen, the large amount of the glycosaminoglycans, also called GAGs, and the multi-adhesive glycoproteins which are present in it. So the type of the collagen which are present in extracellular matrix are mainly the type 2 collagen which is present, but in addition to type 2, type 9, 10 and type 11 are also present. So next are the GAGs, the glycosaminoglycans. These may be sulfated, these may be non-sulfated. The sulfated ones are chondroitin sulfate and keratin sulfate. They are attached to the multi-domain core protein to form proteoglycan monomers called the aggregan. So in this slide you can see these are basically the chondrocytes. The larger fibers, these are those of the collagen which are present in it. These are the type 2 collagen fibers which are present. In green color you can see this thread in green color is hyaluronan which is present, and these smaller are the proteoglycan monomers which are present. These proteoglycan monomers are also called the aggregan. So this is the structure, the gross general structure of the extracellular matrix of the cartilage. This is a detailed diagram in which you can see the aggregan or the proteoglycan monomer. In the center of this structure you can see core protein is present, and to attach the core proteins are the GAGs. And this core protein is attached to the hyaluronan by means of the linker protein. So in this slide you can also see this area contains the proteoglycan. In the center you can see the hyaluronan which is present, and these are the collagen fibers. So how they are connected to each other? Look at this diagram. In this you can see this is the hyaluronan which is present, which is taken over here from this area in the center. Then the linker protein to which core protein is attached, and to this core protein you can see the gags are attached, and background you can see in the dark pink color, this is the collagen type 2 fibers which are present. So this is a schematic representation of most abundant molecules in the cartilage matrix. It shows the interaction between type 2 collagen fibrils and proteoglycans linked to the hyaluronan. The link proteins are non-covalently bound to the protein core of proteoglycans to the linear hyaluronan molecules. The chondroitin sulfate side chains of the proteoglycans are electrostatically bind to the collagen fiber. So this is the detailed structure and the schematic diagram of the extracellular matrix of the collagen of the cartilage. Again I repeat, these are the collagen fibers, type 2 collagen mainly present. Then these threads are the hyaluronan, and to these threads are attached the proteoglycan monomers. This is the detailed structure of proteoglycan monomer, also called aggregan. In this proteoglycan monomer you can see in the center a core protein is present, and to this core protein the glycosaminoglycans are present, which are in case of the cartilage the keratin sulfate and chondroitin sulfate. This core protein monomer is attached to hyaluronan by means of a linker protein. So in this you can see these are the collagen type 2 fiber, these are the proteoglycans, and in the center you can see hyaluronan is present, and again the same structure in detail. Background you can see the type 2 collagen, and these are the proteoglycan monomers. In the center core proteins, the linker protein, and the hyaluronan. So now we discuss the different types of the cartilages. These are the three main types of the cartilages: the hyaline cartilage, the elastic cartilage, and the fibrocartilage. Hyaline cartilage is most commonly occurring. All hyaline cartilages are covered by pericardium except the articular cartilages. So this is again very important MCQ as well. In this slide you can see over here the pericardium is present. Also you can appreciate the pericardium in this location as well. The type of the collagen fibers which are present in extracellular matrix in case of hyaline cartilage are the type 2 collagen fibers, and in addition to this the type 9 and type 11. So these collagen fibers are very thin, and we cannot see these collagen fibers in the light microscopy, and because of the other components of the extracellular matrix, this gives a glossy appearance to the ground substance, to the cartilage, and that is why the name of the hyaline cartilage, it is taken from the word hyalos, and hyalos mean glossy. So it gives a glossy appearance on cross section, and this glossy appearance is because of the reason that we cannot see the type 2 or type 9 or type 11 fibers, which are very thin, and the ground substance which is present, and it contains 60 to 80% amount of the water. That is why it gives a glossy appearance to it. The extracellular matrix in different regions is called the pericellular, the territorial, and interterritorial. The pericellular one is present which is just closer, just in close vicinity to the lacunae, and it contains large amount of the sulfated extracellular matrix. Next to this area contains the territorial matrix, which contains a smaller amount of the type 2 collagen fiber as well. And in interterritorial matrix, large number of the type 2 collagen fibers will be present, and this area is lighter in color as compared to the pericellular or the territorial matrix. So location of the hyaline cartilage, these are again your very important MCQs as well. It is present in epiphyseal cartilage, the laryngeal cartilages, mainly cricoid, thyroid, arytenoid cartilages, in trachea and bronchi, in costal cartilages, in articular cartilages, and certain nasal cartilages are also hyaline in nature. So these are the location of the hyaline cartilage. This is again a very beautiful H&E stain section. This is at the lower magnification. You can see the chondrocytes which are present in the lacunae. In this area you can see a single chondrocyte, and in over here you can see group of 2. At this location you can see 4. Again, very important point, you can notice in this area these asterisks shows that this is the territorial matrix which is stained very dark, and in the section the lacunae and the chondrocyte is not taken in the section. Again, important point is that in living state, these chondrocytes and their lacunae occupy the whole of the space. But when we prepare the slide, they shrink, and because of this, we can see these empty spaces over here. At the periphery, you can see perichondrium, and this is the inner cellular layer of the perichondrium. This is again another slide in which you can see the isogenous group of the chondrocytes which are present, and from this area it is shown over here in higher magnification, in which you can see the chondrocytes in growing area, and also the in the inner cellular layer of the perichondrium, where you can see the active chondrocytes which are present, and these will differentiate into chondroblast. Next is the elastic cartilage. This elastic cartilage is similar to hyaline cartilage, except that it contains the abundant network of elastic fibers in addition to type 2 collagen. So in this slide you can see in the background large number of the elastic fiber which is stained black with the special stains, and you can appreciate this is the slide of the pinna of the ear. Here you can see the epithelium of the pinna which is stratified squamous keratinized type. Then you can see the perichondrium, and here you can see the elastic cartilage, and this elastic cartilage is over here shown in higher magnification. So this elastic cartilage, because of the elastic fibers, gives a yellow color to in a cross section in a freshly cut section. Perichondrium is also present in case of the elastic cartilages. The cell may be present as single or in isogenous groups. Location very important and very easy to remember. The auricle of the ear, walls of external auditory meatus, eustachian tube, epiglottis, and upper respiratory tract. So in all of these locations, this is E, which will help to memorize the location of the elastic cartilage. Next is the fibrocartilage. The fibrocartilage is very tough, yet it is cushioning. It supports the tissue for bone. Chondrocytes of the fibrocartilage occur singly and often in inline. You can see these are the chondrocytes of the cartilages. They are present in as single, and they are aligned in a form of the rows. Type 2 collagen is basically present, and other extracellular cartilage matrix, as I have told you in previous slides. Areas with the chondrocyte and the hyaline matrix are separated by other regions with fibroblasts and dense bundles of type 1 collagen, which confer the extra tensile strength to this tissue. So you can see in this area, these are the area of the cartilage, and these are the area where the type 1 collagen, and the arrows show presence of the fibroblast over here. So fibrocartilage along with the type 1 collagen and the fibroblast, so this gives extra tensile strength to this tissue. The relative scarcity of the proteoglycans overall makes the fibrocartilage matrix more acidophilic. So you can appreciate this change, and you can appreciate it. You can differentiate it from the previous slide, that in this the territorial matrix is little bit acidophilic, and this is because of the scarcity of the proteoglycans which are present. Again, very important point, there is no perichondrium in the in case of the fibrocartilage. So now the location of the fibrocartilage, it is located at intervertebral disc in attachment of certain ligaments, symphysis pubis, and in the menisci. So again you can see this is the lower magnification of the fibrocartilage, in which you can see the chondrocytes which are present in a form of the rows. Here you can see these are the present in the form of the rows, and in between these chondrocytes you can see large amount of the type 1 collagen which is present. So these are the three type of the cartilage. You can easily differentiate between these. This one is the hyaline cartilage. The cells may be present in the form of this isogenous group or maybe singly. It gives a glossy appearance, and the territorial matrix usually stain dark basophilia because of the sulfated gags which are present over here. In this case, this is the slide of the elastic cartilage, and in this you can see on the background the large number of the elastic fibers which are present over here. These fibers usually stain black. In many slides also the cells may be present as singly or in double. It also has the pericardium. And this is slide of the fibrocartilage, in which you can see the cells which are present in the form of the rows over here, and in between the chondrocytes you can see the type 1 collagen which is present. So this is all for the today's class. The references for the today's class are Basic Histology by Laik Hussain Siddiq, the Histology text and atlas by Jan Quera, Histology text and atlas by H. Ross. Thank you very much for your attention, and Allah Hafiz till the next class.